we develop theoretical models, and we also analyze data and develop statistics, but ranging from across biological scales, including, for example, thinking about hybridization, adaptation to challenging environments, but also sometimes we're concerned about the individual effects of single mutations. And while these are all wonders of evolution, they can also sometimes be express themselves as curses of evolution. And so today specifically, I want to talk about virus adaptation to a challenging environment. And many of you might be aware that RNA viruses have particularly high mutation rates in as compared with other organisms, as seen here. Like compared with their genome size, their mutation rates are really high. So the RNA viruses are here the red pointers. And that's why it is assumed that mutagenic drugs might be able to kill RNA viruses. And there's this mutagenic drug favipiravir, which interferes with the RNA dependent polymerase of the virus, and which seems to looks quite promising for use against RNA viruses in general, and it has already been approved in Japan, maybe in the US too, and it has been used also against other RNA viruses. In fact, it's also in trials against COVID at the moment, but I don't think it looks very good so far. And of course, that also led to many studies that showed that tried to find potential resistance mutations in the lab. And there's surprisingly little seen so far in the literature, except maybe for this paper here, which I'm going to come back to also in a little bit. So the way that these mutagenic drugs are supposedly killing the population is really a fundamentally evolutionary principle, which has been named both mutational meltdown and lethal mutagenesis in the literature. And this is all based on this theory of Maller's ratchet, which refers to the stepwise loss of the fittest genotype due to the accumulation of deleterious mutations in asexual populations. And so the idea is that the virus population just accumulates more and more mutations. Most of them will be deleterious. And then as soon as once the population mean fitness or the growth rate falls below 1, then the population will be driven to extinction. And it has been shown in plenty of different theoretical studies that this is can either be caused by just mutation pressure due to high mutation rates, but also just by small population sizes when purging is less efficient. And there's really a very rich body of theory about these different kinds of things. So during my postdoc, I got involved in a study of influenza in the lab, where we found that actually this drug favipiravir can in some cases kill a virus population. So in this plot here, you see time on the x-axis measured in viral passages in the lab. And then these colorful lines, they represent allele frequency trajectories from deep sequencing. And then this black line represents some estimate of the effective population size. And as you can see, after some time, there's a lot of accumulation of mutations, but also there is extinction at the end to basically levels where it wasn't possible anymore to passage the virus any further. And so this leads to the first question that I want to talk about today, which I'm not sure she said exactly these words, but the project that Lucy Lange-Houston, who is also here in the audience, wanted to tackle was to figure out how long the time to mutational meltdown is in a population that is under these pressures. And the classical theory has already basically predicted, especially in this Lynch paper here, that there are 3 phases in the evolutionary dynamics going from an expanding population to mutational meltdown. So time is again here on the x-axis, and then we have two lines here. The blue one is the population size, and the grayish one is the number of mutations that accumulate. And so what you can see is that initially there's an expansion of the population, and just a relatively deterministic accumulation of mutations until some time when the redshift begins to click. And then from that time on, we have this almost linear clicking of the redshift at some rate until the mean fitness of the population falls below 1. And then again, we have some rather deterministic decline of the population towards extinction. And so these three phases are basically, so basically we have like these deterministic phases at the beginning and the end, and then this redshift phase in between. And what Lucy did was to basically put all three together and try to just sum them up in order to calculate the extinction time under mutational meltdown. And so basically that's what it looks like. It's not very pretty, but it's basically just summing up those three different phases. And I mean, I think that the one of the challenges here is that in any case, we need to calculate the redshift speed. And so those of you who are familiar with the literature, they might know plenty of references to different ways of calculating the redshift. And interestingly, they don't seem to do well in our desired parameter range. And what Lucy found out was that currently we're working with this estimation here from Gessler, which seems to do best. So, but one thing that I would be really interested in from this audience would be to maybe give us some additional ideas how to compute the redshift speed during this phase. And so basically, so what we're describing is the first this phase where the population expands and goes towards mutation selection equilibrium. Then once the frequency of the class that has 0 mutations drops below 1, we get into the redshift phase. And then once the mean fitness drops below 1, we get into the mutational meltdown phase, which ends once the expected population size is below 1. And well, you can see here roughly how this analytical prediction performs. On the left side, you see some simulations, and on the right side, the analytical estimation, where there's just some area where it's not possible to calculate it. And you can see that on average, it goes quite well, but sadly, exactly one parameter range that we would probably be quite interested in, namely here, those relatively small selection coefficients and high mutation rates, they don't work so well. And this is really due to the redshift speed accumulation approximation, which doesn't hold very well at that point. So basically, to sum this up, this first part is, so we found this analytical approximation of the extinction time, which captures important features of mutational meltdown. But computing the speed of the redshift is actually a challenge, and that's quite, it was quite surprising considering how much literature there is actually on the speed of the redshift, how much of a challenge it would be to find some way of using this. So next, I mean, we found that in some cases the population was dying from the drug, but not in all cases. For example, here is another treatment from this paper that I showed you before. And here the drug concentration wasn't increased all the time, but it was kept constant at some point. And even though you see a lot of mutations segregating in this in the dynamics here, the population did not go extinct even until the end of the experiment. And so it seems that maybe there is some potential for the population to adapt to the drug. And so that's kind of what intrigued me somehow after working on these data. And so I started thinking about by which mechanisms could actually the virus adapt to mutagenic drugs. And for this, I just want to make one comment, namely that once I started working at IGC, and I talked a lot with people who are interested in different kinds of resistance. And so I learned that evolution of resistance is a type of adaptation, but not all adaptation mechanisms should be always called resistance. And so let me explain this in the next slide, because the different mechanisms I thought about are the following. So firstly, you could just get a continuous kind of runaway, like where the virus would just get improve its growth and thereby somehow escape the death. And there's also some quite nice theory about this already developed. And but that's kind of just it has to go on all the time. So it's not a real kind of resistance forever. Then secondly, you might get something like a mutation rate modifier, and that would really just be something that returns the mutation rate of the virus back to something that's that it was before, without the mutagenic drug, and that's what I think would qualify as resistance, because it's like it really brings the population to its old state in the presence of the drug. And then this, the third thing I could think about was this so-called modifier of the distribution of fitness effects. And that could be something, mutation that changes mutational effects themselves, and that could be possibly genome-wide. And by doing so, it could lead to something that would rather qualify as drug tolerance, because the mutation rate would still be just as high, but this drug would be tolerated by the virus and it would not go extinct. And so I want to go more into this third mechanism and just explain to you how this looks. So if you think of the deleterious selection coefficient here in these simulations and the time to extinction, then Maller's redshift has this is specifically interesting because it clicks the fastest for intermediate selection coefficients, which means that, no, it doesn't click the fastest, but the time to extinction is has a minimum for intermediate selection coefficients. So that means that if we actually have selection coefficients in this area, then we can improve or like slow down the redshift, both by moving, by making more deleterious mutations, or by making all mutations more deleterious, and by making them more beneficial. That is, in this case here, they just don't matter so much anymore. So it's a gradual process where it's just kind of some buffering. And then on the deleterious side, it's like by increasing the mutational effects, you improve the efficiency of purging of the mutations. And so that's kind of how our distribution of fitness effect modifier could slow down the redshift. And that led us to just address with simulations how likely it is that there is an invasion of this distribution of fitness effect modifier. And what Mark in the lab did was to simulate this, and he had to simulate it very often because, as you can imagine, if you suddenly make all your mutations more deleterious, you also make those carriers of the mutations die much more frequently. But if they cross some invasion threshold, then they suddenly can rescue the population altogether. So there are cases in which even if you make mutations much more deleterious, you can somehow develop this tolerance to the drug here. And on the other hand, of course, if you have buffering mutations that somehow improve just the effects of all deleterious mutations, then it's very likely that you will increase the time to extinction. And so to sum this up, we can have theoretically both positive and negative DFE modifiers that can invade, and they can increase the time to extinction of the population. And if we think of this one invasion, the modifier that makes mutations more deleterious, its invasion is really rare, but potentially impactful. And specifically, if you think of lab experiments, most likely you won't catch those, right? Because it really has to happen 100,000 times until you can get one of those to invade. And so that basically leads to the last question that Ana Morales, a postdoc in the lab, is asking. So she's actually asking, by which mechanisms does the virus now really adapt to mutagenic drugs? And she's doing that in the lab using laboratory evolution experiments. And basically, she will be using a rather common approach where that has been used, that is commonly used, where she seeds a number of viruses in a petri dish, then she grows those, and then we can take some samples for sequencing if we're interested, and seed the next passage afterwards. And in any case, so the nice part about this approach is that we can monitor various aspects of the evolutionary dynamics, for example, the bottleneck sizes, absolute growth rates, genome-wide allele frequencies, and other factors that might influence the outcome. And what we specifically want to do now is, because we're interested in these things that probably don't have a very high invasion probability, we do not want to just wait for adaptations to occur, but to study the evolutionary dynamics of strains that carry pre-identified target mutations, which we think could play a role for adaptation to favipiravir. And so basically, then we can come up with different expectations. And given certain strains here on the left side in both the control environment and the drug environment. And if you think about it, so obviously the wild type strain should survive well in the control environment and die in the drug environment. And now if you think of a mutation rate modifier, you should see less segregating mutations in the control environment, whereas it should live longer than the wild type in the drug environment, and then have like rather similar numbers of mutations that you see. And now if you think of those DFE modifiers, you will see for these buffering mutations, you will see that it should survive in both the control and the drug environment, or at least live longer than the wild type. But at the same time, it will still accumulate many mutations, but it just doesn't die because it can now tolerate those mutations. And then for the negative DFE modifier, we would expect that there's less mutation accumulation, even though the same mutations appear, because they are purged much more efficiently. And so it should survive in the drug environment, but accumulate fewer mutations. And so we're going to go then through some kind of cascade of measurements, where the first step will just be to see, okay, can they actually survive longer, and what is their absolute fitness in those treatments? And then if we find anything interesting, we can take samples and do sequencing to also identify allele frequencies and see if anything happened during the experiment. And then ultimately, of course, it would be interesting to then test if they also, if our predictions about the competitive fitness under different starting frequencies are true. And we're currently developing this with some candidate mutations, including those known adaptations to favipiravir, for which their mechanism is also not absolutely clear yet, and also some other proposed mutations from our collaborators' lab. And yes, I would be really happy to also hear your ideas, which other mutations we could test, or what you think hopefully not go too wrong. So what we're doing is to develop experiments to confirm adaptation mechanisms of known and proposed candidates. And in the best case, we will confirm novel adaptations based on these theoretical considerations and known molecular mechanisms. And in the worst case, if that doesn't come out, then at least we can characterize evolutionary dynamics and maybe still in the process find some new adaptations that occurred during the evolution experiment. So to sum up what I was talking about, I think that adaptation to mutagenic conditions is a fascinating evolutionary problem with many open questions. What we did was to sum over these 3 phases of the evolutionary dynamics and approximate the extinction time as first step of solving this analytically. And then we identified different adaptation mechanisms which could play a role here. And these we want to now test whether they actually exist with some candidate mutations. So please stay tuned, or also tune into the discussion during this workshop. And with this, I want to thank you for your attention, and also thank the people that have been involved in this question, in these projects, especially Lucy, Anna, and Mark, and also my virologist collaborator, Maria João at IGC. Thanks.
So thank you very much, Claudia, for the great talk. So please all feel free to write your questions in the chat. I'm going to start reading the questions. So we had a question by Daniel Weisman, who asked, there is a, she said, we've talked about this before, but I think there is a real difference between mutational meltdown and lethal mutagenesis. The latter doesn't rely on Muller's ratchet.
I disagree.
Okay, so Daniel, you're welcome to unmute yourself for the discussion.
Okay, sure, yeah, and Matteo kind of followed up on this, kind of clarifying what I was talking about, but basically, I mean, in your classic lethal mutagenesis, that's just saying you're hitting them with so many mutations that each wild type virus is not producing on average a wild type virus to replace itself. So like really mechanistically, like if it's mutational meltdown and you crank up the fitness effects of mutations, you make them stronger, you make them worse, you're not going to save your population, right? Whereas if it is,
well, I mean, the thing is,
mutational meltdown Muller's ratchet, you will. So that's a real difference in those two cases.
So in the case of lethal mutagenesis, as far as I remember, the